Imagine you're a detective trying to catch this highly dangerous, shape-shifting fugitive. And this fugitive doesn't just change their disguise to slip past the security cameras. When things get really heated, they hijack a police car, convince the local SWAT team to act as their personal bodyguards, and then build this custom armored vehicle just to escape the city limits.
It sounds like, a summer blockbuster. But biologically, it is a disturbingly accurate description of how cancer spreads through the body. We naturally want to think of a tumor as just a static growing lump of rogue cells.
Just a mass sitting there.
Exactly. But when you look closely at the mechanics, especially during metastasis, when it spreads, it behaves much more like an organized, evolving, and deeply deceptive ecosystem.
Which brings us to today's deep dive into a massive, groundbreaking 2026 study from Cell Reports Medicine. It focuses on high-grade serous ovarian cancer, or HGSOC. And to catch this particular fugitive, researchers didn't just look at a few cells in a petri dish.
No, they didn't. They mapped out over 2.5 million microscopic locations within patient tissue. They used multi-omic profiling of over 60 whole genome sequences and 100 single-cell samples. They wanted to figure out exactly how this armored vehicle is built and how it travels.
And the resolution of the data they gathered it's truly unprecedented. And for you listening, understanding this fundamentally changes how we view cancer. We are no longer looking at a disease of just uncontrolled growth.
Right. We're looking at a highly sophisticated travel structure, one that actively corrupts your own body's cells to survive the journey from one organ to another. Because if we can finally understand how this biological vehicle is engineered, we can, learn how to dismantle it.
Okay, let's unpack this. Before we can even get into this wild armored vehicle the tumor eventually builds, we have to understand how this fugitive sneaks out of the original crime scene in the ovary.
Well, to survive long enough to spread, the tumor first has to survive your body's immune defenses. Specifically, it has to evade the T cells, which act as, they're the cellular police force constantly patrolling your tissue.
Patrolling for anything that shouldn't be there.
Precisely. So the researchers started by looking at a concept called PTMB, which stands for persistent tumor mutation burden. Now, logically, as a tumor grows, becomes more aggressive, and metastasizes to a new site like the omentum, which is a fatty layer in your belly, you would expect it to accumulate a massive amount of mutations.
Right. You'd think an aggressively spreading tumor would be riddled with this chaotic mess of genetic errors compared to the original primary tumor.
I mean, it's becoming more advanced, so it should be mathematically more complex, right?
You'd think so. But the data showed the exact opposite. The tumors that successfully made the journey and metastasized to the omentum actually had the lowest persistent mutation burden.
Wait, the lowest?
Yeah, the lowest. They'd actively dropped mutations, making themselves mathematically simpler than the primary tumor. That seems entirely backward. Why would dropping mutations help a cancer cell survive?
Because mutations are a double-edged sword. While they drive the uncontrolled growth that cancer needs, they also create these byproducts called neoantigens.
Neoantigens, let's break that down.
Sure. Think of neoantigens as little abnormal proteins that pop up on the outside of the cancer cell. To a passing T cell, those abnormal proteins act like bright red flags. They signal that the cell is damaged or hostile.
Oh, I see.
So if a cancer cell holds on to all its persistent mutations, it's basically walking around waving a giant neon sign that says, I am an invader, destroy me.
So to evade the police, the fugitive just takes off the bright red shirt. But how do they actually drop the mutations?
Usually through massive chromosomal deletions. The cancer cell literally deletes entire chunks of its own DNA. It just throws away the genetic code that produces those specific red flags.
That is wild.
It is. But dropping the mutation burden is only the first step. The study identified a second, much more drastic hiding technique called HLA-LOH, or loss of heterozygosity of human leukocyte antigen.
Okay, let's break down that acronym because that's a mouthful. What is the HLA actually doing day to day in a normal cell?
Right, so HLA molecules function as the ID badges or window displays of the cellular world. Every healthy cell in your body uses the HLA complex to take a tiny sample of the proteins it's producing inside, and it displays them on the outside.
So it's showing the immune system what it's working on.
Exactly. It's how a cell proves everything is functioning normally. When a T cell patrols your tissue, it looks in that window. If it sees healthy proteins, it moves on. If it sees a neoantigen, one of those mutated cancer proteins, it attacks and kills the cell.
So if the cancer cell undergoes this HLA-LH process, what happens to the window display?
It's just gone. The tumor deletes the actual genetic code required to build those ID badges in the first place.
Wow, so it's not just taking off their red shirt, they wipe their fingerprints from the database entirely. If they don't have an ID badge to check, does this mean the T cells just walk right past them?
Essentially, yes. If the cell physically lacks the HLA structure to present its internal proteins, the T cells bump into it, find nothing to read, and move on. They are completely unaware that they just bypassed a highly lethal cancer cell.
It's a brilliant, terrifying, evolutionary disappearing act. So just to make sure I'm wrapping my head around this and to summarize this first part, cancer isn't just growing mindlessly, it's actively deleting parts of its own identity, stripping away its mutations and its ID badges just to survive the immune system.
It completely flips the traditional narrative of cancer just blindly accumulating damage.
It really does. So for you listening, have you ever thought about cancer not as an uncontrollable growth, but as a population actively deleting its own traits just to survive? Shedding a trait as the ultimate survival move.
It's a profound shift in perspective.
But wait, if the tumor is wiping its fingerprints and making itself completely invisible to the immune system, it should be invisible to our lab microscopes too. How do the researchers actually track these specific cells if they dumped their ID badges?
Well, they use a technological marvel developed for this study called STARLETS, which stands for Spatiotemporal Tumor Clone Evolution Tracking System.
Spatiotemporal, so space and time.
Exactly. To appreciate why this is such a game changer, you have to look at the old way we studied tumors. Historically, we take a biopsy, grind the tissue up, and run RNA sequencing.
Like making a smoothie.
Right. If you picture a fruit bowl, the old method is like throwing the whole bowl into a blender. You can analyze the smoothie and know there were strawberries and bananas in there, but you have absolutely no idea which strawberry was sitting next to which banana before you blended it.
You lose the map entirely. You just have a list of ingredients.
Precisely. STARLETS keeps the map intact. It harmonizes whole genome sequencing with something called spatial transcriptomics.
And spatial transcriptomics does what exactly?
It allows scientists to look at a microscopic, intact slice of tissue and map exactly where specific individual tumor clones are located. More importantly, it shows us exactly which of the patient's own cells, like immune cells or structural cells, are sitting right next door to the cancer.
So we can see the neighborhood, we can see exactly who the fugitive is interacting with.
Yeah. And by mapping out those cellular neighborhoods over time, STARLETS revealed that the tumor has to survive an incredibly harsh two-step Darwinian gauntlet just to escape the ovary.
Walk me through this gauntlet. What is the first hurdle?
Step 1 is hypoxia selection. Deep inside the core of the primary tumor, the cancer cells are reproducing so incredibly fast that they outgrow their own blood supply. They literally deplete the available oxygen in their microenvironment.
So they're suffocating.
Yes. This state of oxygen starvation is called hypoxia. The sheer stress of suffocating induces severe DNA damage, and a massive number of cancer cells simply die. But the ones that survive undergo a chemical shift. The hypoxia triggers a migration response.
So it's not a coordinated tactical march. It's panic. It's like a crowded room running out of air, and the survivors are desperately pushing their way outward to the edges of the tumor just to breathe.
That's exactly the dynamic. But reaching the edge brings them to step 2, immune selection. As these surviving cells push out to the tumor stromal boundary, the very edge of the tumor, they run headfirst into the patient's immune system.
Out of the frying pan and into the fire.
Literally. The T cells are waiting for them at the border. It is an absolute slaughter. The T cells wipe out almost every cancer cell that tries to cross.
Let me guess, the only ones that survive this border crossing are the ones that went through that HLA-LOH process, the invisible ones.
You nail it. The T cells annihilate every cancer cell that still has its ID badge intact. The only cells that make it through the massacre are those specific mutated clones that deleted their HLA complex.
Because they lack the window display.
Right. The T cells miss them. And these elite survivors fall out of the ovary and drop into the abdominal cavity. And the researchers even proved this mechanism in live mice. They used CRISPR-Cas9 technology to artificially knock out the mouse equivalent of the HLA gene.
Oh, wow. So they made them invisible on purpose.
Yes, to test the theory. When they subjected those tumors to a T-cell attack, the normal cancer cells were eradicated. but the invisible knockout clones selectively survived and multiplied.
Here's where it gets really interesting. Wait, so your body's own defense system is accidentally acting as an elite training camp. By slaughtering all the weak, visible cells, the T cells are literally ensuring that only the most invisible, dangerous supervillains make it out into the abdomen.
That is the dark irony of Darwinian selection in oncology. The relentless pressure of your own immune system is what forges the perfect metastatic cell.
Think about your own immune system. We're taught from childhood that our white blood cells are the ultimate protectors. But to summarize this section, in this specific microscopic gauntlet, first suffocating from hypoxia, then facing immune selection, that defensive pressure is exactly what trains the cancer to become an unstoppable fugitive.
It's terrifying, but also incredibly elegant in a twisted way.
So to you listening, how does it change your view of your body's immune system knowing its pressure actually forges stronger metastatic cells? It's a lot to process, but dropping into the abdominal fluid, would you call it?
Ascites. The fluid is called ascites. But dropping into it is only the beginning of the journey. Single floating cancer cells are highly vulnerable. They need to travel across the abdomen to implant on other organs like the liver or the omentum.
So they need a vehicle.
Exactly. To survive that voyage, they build a microscopic spaceship. The researchers named it the tripartite ensemble.
Tripartite meaning 3 parts. So how do the researchers actually see this microscopic spaceship? Because tracking a single cell in fluid seems impossible.
It is difficult, but they used ultra-high-resolution 3D imaging. They applied a chemical clearing technique called UDisco, which literally strips the fats and lipids away from the tissue.
Strips the fat away.
Yeah, it turns the tissue completely glass clear. Then they paired that with something called second harmonic generation, or SHG, which specifically lights up collagen fibers. This allowed lasers to map the specific proteins inside without having to slice the tissue up.
So they get a perfect 3D picture, and what emerged...
What emerged in the 3D rendering were these perfect armored spheres floating in the abdominal fluid.
Okay, so what did the lasers actually see? Break down these 3 layers for me.
Let's start at the very center, the core of the sphere. You don't find cancer cells there. You find SPP1 positive macrophages.
Macrophages. But those are white blood cells. Those are our immune cells. Why are they at the center of a cancer vehicle?
They've been corrupted. Normally, macrophages act as the garbage collectors and tissue repairmen of the immune system. They clean up cellular debris and help heal wounds.
But the tumor gets to them.
The tumor biochemically brainwashes them. It forces them into a specific state where they secrete a protein called SPP1. In this corrupted state, the macrophages act as the engine and the organizers of the metastasis.
Unbelievable.
Yeah, they release signals that promote blood vessel growth and actively prepare the new organ to receive the tumor.
So the corrupted immune cells are running the engine. Where is the actual cancer?
The cancer is the middle layer. The malignant epithelial cells securely wrap themselves around that macrophage core. They are safely tucked away, riding entirely as passengers.
Which brings us to the third, outer layer, the armor.
The outer shield is constructed entirely of MMP11 positive myCAFs. That stands for myofibroblastic cancer-associated fibroblasts.
Fibroblasts, the builders.
Right. Normally, fibroblasts are the structural builders of your body. They lay down connective tissue and collagen to close cuts and heal injuries. But the tumor hijacks them, forcing them to lay down a thick, dense matrix of collagen completely surrounding the sphere.
And the second harmonic generation lasers saw this collagen.
Exactly. This collagen matrix acts as a physical, bulletproof shield. T cells literally cannot penetrate it to reach the cancer cells inside.
Let's stick to our fugitive analogy because it perfectly maps out the mechanics here. This isn't just a cell floating away. This is a biological VIP motorcade.
It really is.
The macrophages are the hijacked drivers running the engine and navigating the route. The cancer cells are the VIPs sitting comfortably in the back seat. And those hijacked fibroblasts are acting as the heavy, bulletproof glass wrapped around the entire convoy.
What's fascinating here is the sheer audacity of the mechanism. The cancer doesn't expend its own energy to build the armor or drive the vehicle. It utilizes the host's own biology to survive.
It recruits your body's cells to do the heavy lifting.
Exactly. And this specific structure, this tripartite ensemble, is what floats safely through the fluid, lands on a healthy organ, and seeds a brand new tumor.
So just to do a quick recap, we have an armored sphere with a corrupted immune cell core, cancer cell passengers, and a bulletproof collagen shield. So, for you listening, can you picture this microscopic armored sphere floating in the abdominal fluid? Which of the 3 layers do you think is the weakest link? Because if I'm looking at this, I want to take out the engine, I want to take out the drivers, but how do we get past that bulletproof collagen glass?
You don't try to break the glass. You dissolve the vehicle from the inside out by targeting the biochemical glue holding it all together.
The glue!
Yeah. The researchers discovered that this entire 3D structure only stays intact because of a highly specific communication network between the corrupted macrophages in the core and the cancer cells in the middle. They call it the SPP1-CD44 axis.
Let's break down the mechanics of that axis. What exactly is this glue?
Well, remember that SPP1 protein. It's the signal constantly secreted by those corrupted macrophages driving the car. And CD44 is a receptor protein sitting on the surface of the cancer cells.
So a key and a lock.
Precisely. When the SPP1 protein physically slots into the CD44 receptor, it chemically commands the cells to stick together, grow, and maintain that outer armor. It is the molecular glue that keeps the motorcade in tight formation.
So it's the radio communication between the drivers and the VIP passengers. Cut the wire, and the convoy stops.
Exactly. The researchers tested this vulnerability in 2 ways. First, they used a drug called angstrom-6, or A6. A6 is a CD44 antagonist.
Meaning it blocks it.
Right. Its mechanism is simple. It physically plugs up the CD44 receptor on the cancer cell, meaning the SPP1 signal from the macrophage simply bounces off. It can't land. Second, they used a neutralizing antibody designed to hunt down and neutralize the SPP1 protein in the fluid before it ever reaches the cancer cells.
And what happened when they cut that communication line? Did the convoy stop?
In both laboratory dishes and in live breathing mouse models, they used ID8 tumors in the mice. The results were dramatic. Blocking this axis caused the armored spheres to physically fall apart. The spheroids disaggregated.
They just dissolved.
Yeah. Without the glue telling them to stick together, the structure collapses, the cancer cells lose their protective collagen shield, and the overall tumor burden in the mice was visibly and significantly reduced.
That is incredible. They found the single point of failure in the entire motorcade. But a mouse model is one thing. Do we actually see this vulnerability holding true in human patients?
We do, and this is where the clinical data gets highly counterintuitive, but incredibly hopeful. The researchers went back and analyzed real-world human data from 2 previously conducted clinical trials, the Option S-01 trial and the NANT trial.
What were the mechanics of those specific trials? What treatments were the patients actually receiving?
In the Option 01 trial, patients were treated with an oncolytic virus called H101.
An oncolytic virus?
Yeah, an oncolytic virus is a virus genetically engineered in a lab to specifically infect, replicate inside, and kill cancer cells while ignoring healthy tissue. They were injecting this virus directly into the patient's abdominal fluid. The NANT trial, on the other hand, was testing PRP inhibitors.
Let's drill down on that. Remind me how a PRP inhibitor works on a cellular level.
PRP is an enzyme that cancer cells heavily rely on to repair daily damage to their own DNA. A PRP inhibitor blocks that enzyme. Without the ability to repair itself, the cancer cell's DNA essentially shatters and the cell dies.
Got it. So you have 2 very advanced, very different therapies.
Right. And when the researchers analyzed the tissue of the patients in these trials, they looked specifically at their baseline levels of those corrupted SPP1 positive macrophages.
The drivers, the engines of the convoy. So logic would dictate that if a patient has a massive amount of those corrupted macrophages, their cancer is building more motorcades, spreading faster, and they should have a much worse outcome, right?
That is exactly what you would assume. But the clinical data proved the exact opposite. The patients who had a higher baseline abundance of these bad SPP1 positive macrophages actually responded significantly better to the therapies.
Wait, really? Better?
Much better. They had longer times before their abdominal fluid needed to be drained and showed much stronger responses to both the virus and the PRP inhibitors.
Hold on, that doesn't make any sense. If those corrupted macrophages are the engine driving the metastasis, how does having more of them suddenly become a good thing when you introduce the virus?
If we connect this to the bigger picture, it makes perfect biological sense. The researchers discovered a fatal flaw in the tumor's design. Those specific SPP1 positive macrophages happen to express very high levels of 2 specific receptors on their surface, called CXADR and ITGAV.
CXADR and ITGAV.
Okay. You can think of these receptors as molecular docking stations. And it just so happens that those exact docking stations are the main entry doors that the H101 virus uses to breach a cell.
Oh, wow. I see where this is going.
By forcing the patient's body to produce millions of corrupted macrophages to build its armored convoys, the tumor is inadvertently building millions of perfectly tailored docking stations for the virus.
So it's literally inviting the virus in.
Yes. The therapy virus enters the fluid, spots the macrophages, latches onto the docking stations, and moves inside. The virus essentially infects the driver, blows up the engine of the convoy, and the entire metastatic structure collapses.
It's the ultimate Trojan horse. The cancer thinks it's recruiting your immune cells to build an impenetrable shield, but we are using the very cells that make up that shield as the back door to completely destroy it.
Exactly. It takes the tumor's most sophisticated survival mechanism and turns it into a giant virus-sized bullseye.
So to recap this final segment, we dissolve the glue, the SPP1-CD44 axis, and the motorcade falls apart. And counterintuitively, having more of those corrupted immune cells actually makes virus therapies more effective because they act as docking stations. For you listening, isn't it incredible that discovering the tumor's best armor is exactly what handed us the blueprint to defeat it?
That's a huge leap forward.
So what does this all mean? Let's zoom all the way out. We started by tracking a fugitive that wiped its genetic fingerprints, literally deleting its mutations and its HLA ID badges, just to survive the panic of oxygen starvation and the brutal gauntlet of your immune system's T cells.
Then we watch those few elite surviving cells drop into the abdomen and manipulate the host's own biology to build the tripartite ensemble, a highly organized, three-layered armored convoy driven by your corrupted macrophages and shielded by hijacked tissue builders.
And finally, thanks to the incredibly precise mapping power of STARLETS, we didn't just passively observe this vehicle. We found the chemical glue holding the convoy together, and we saw real human clinical data proving that we can use the tumor's own armor as a targeted entry point for oncolytic viruses and advanced drugs.
Which leaves us with a final provocative thought to take with you. For decades, the entire field of oncology has been obsessively focused on developing drugs that strictly target and kill the cancer cell itself.
Right, trying to snipe the cancer.
Exactly. We've spent billions of dollars trying to shoot the passenger. But if tumors rely so heavily on our own corrupted cells, like these macrophages and fibroblasts, to build these intricate travel convoys, and if we know this specific structure exists across multiple types of cancers,
could the future of curing cancer be less about attacking the passenger and entirely about blowing up the vehicle?
That's the question we have to ask ourselves moving forward.
Because if you take away the armored car, the shape-shifting fugitive doesn't stand a chance.